All right, welcome to chapter 12 on eukaryotes. The different types of eukaryotic cells we're going to be talking about in this presentation are on fungi, algae, protozoa, helminths, and we're going to talk a little bit about arthropods at the very end. So to get us started, we're going to talk about fungi. So fungi commonly grow in acidic, low moisture, and high osmotic pressure environments. What I just described to you is the environment of the skin, right? So this is where we're going to be finding fungi quite commonly, is on the skin. The cell walls of fungi are made of chitin. It's important to note here that not just for fungi, but for all the eukaryotic cells that we're talking about, actually for most of what we're talking about in this entire class, we tend to focus a lot on the pathogenic species because this is catered towards human microbiology. But do note here that actually most fungi and most species in general are not pathogenic towards humans. So they're actually, quite a lot of them are beneficial or have no effect on humans. So do keep that in mind. So most fungi are actually decomposers. So it's very important for the environment. But all fungi absorb and metabolize complex carbohydrates, so they are chemoheterotrophs, and only a few are pathogenic to plants and animals. Now there's two types of fungi. There's molds and yeasts. So the molds are on this side. So the molds they have these filaments. These filaments are where the cells have connected to each other. They end up being multicellular. And they have these filaments. They're like finger-like projections. Whereas the yeasts are all unicellular. We all have these individual cells independent of one another. And those are yeasts. So you would say yeasts are non-filamentous. Another difference between molds and yeasts is whether they're aerobic or not. So molds on this side are aerobic, whereas yeasts are facultatively anaerobic. Facultatively anaerobic species, remember, can undergo aerobic and anaerobic respiration. Other species are what we call dimorphic. So they can be a little bit more yeast-like at body temperature and more mold-like at room temperature. The life cycle of mold. So here we're talking about mold. Remember, molds have these filaments. The filaments we're going to call are hyphae. Okay, so hyphae are these finger-like projections. Just keep that in mind. We're talking about molds. Molds are the ones that have the filaments, okay? They have this life cycle. So in the mold life cycle, they're going to produce spores. So down here, they undergo the process of sporulation, which is the production of spores. They're going to do this when they're in a healthy environment. They're actively growing and reproducing, and they're going to make spores. And those spores will then be released into the environment. If you remember back to when we were talking about bacteria, prokaryotes, where we have the production of endospores, and the endospores are going to allow that bacterial species to survive in a harsh environment. Here it's a very similar concept. So we have these molds are going to produce spores, but in this case, the spores are a form of reproduction. So this is how these mold are reproducing. They're producing these spores, and the spores are going to be spread out, and then they're going to germinate when they reach a favorable environment, and then go back to being a mold with the filaments. So some examples of fungi. So Saccharomyces cerevisiae help us to produce bread, wine, and also vaccines. Taxomyces produces Taxol, which is a cancer drug. And Paecilomyces helps to kill termites, so we use that to our advantage as well. Now, there's also some disadvantages of some fungi. So we've probably all been familiar with moldy bread. Okay, so fungi is responsible for a lot of food spoilage, which can be economically detrimental. And it's also responsible for causing several different diseases. So here's a lot of different diseases. Don't worry too much about memorizing all of these. It's just for your curiosity. What I do want you to focus on is the different mycoses. So mycoses means a fungal infection, and there's several different types of fungal infections. So I do want you to know these. So we have systemic mycosis. That's going to be a fungal infection that's systemic, right? It's throughout the entire body. So this is going to be deep within the body, not just on the skin, but deep in the body. It's going to affect all the tissues as well as the organs. The next type is subcutaneous mycosis. So it's a localized infection, but it reaches all the way down into the subcutaneous. So it goes through the entire skin layer to slightly below the skin. What we also see a lot of fungal infections are opportunistic fungal infections or opportunistic mycosis. Because the body is able to fight off most fungal infections if we have a healthy immune system. We are exposed to fungus constantly, and our immune system usually can fight that off. But if we have a weakened immune system, then these fungal infections can overcome the immune system. So we see this a lot in compromised hosts, patients with a weakened immune system, AIDS patients. We see a lot of these opportunistic mycoses. Candidiasis is a big one. And usually these can become systemic. More mycoses. We have cutaneous mycosis. So that's going to affect anything that has keratin. So any keratin-containing tissues. It's going to include hair, nails, and skin. So the candidiasis that we just talked about with the opportunistic infections, that's a cutaneous mycosis. Ringworms down here, that's another really common one. And then lastly, we have superficial mycosis. So this is going to be even more superficial than cutaneous. So it only is going to affect the hair shaft and the superficial skin cells. So if you remember that the superficial layers of the epidermis are not living, those cells eventually die because they get too far away from the blood stream, the nutrients from the bloodstream. So because superficial mycosis is not affecting any living cells, it's not going to cause an immune response from the host. So that was all fungi. Now we're going to move on to algae. So we don't have any algae that directly infect humans. So for the most part, algae are not pathogenic, but there's a couple of examples that we're going to talk about on how their toxins can cause disease. But before we get into that, a little bit about algae itself. So algae are photoautotrophs. So they undergo photosynthesis. And we usually find these in aquatic environments. So algae is the bottom of the food chain in like the ocean, right? So this is what supports all life in the ocean is algae. They reproduce asexually, but there are some that some multicellular algae that can produce sexually via alternation of generations. There's lots of benefits of algae. So they fix carbon dioxide into organic molecules. They produce 80% of the Earth's oxygen, so it's huge for the environment. They produce oil, and they're also symbionts of many animals. But we're going to talk about 2 examples of how they can be pathogenic. And again, it's not that the algae themselves are causing disease, but rather it's that they're producing toxins that are toxic. Okay, so the first example is diatoms. Diatoms produce diamoic acid. This is a neurological toxin, so it's going to cause lethargy, disorientation, seizures, and death. And the way infection occurs is through ingestion of mussels that have fed on those toxic diatoms. So if a mussel has fed on those diatoms, then it's going to have the toxins inside of it. And then if we eat those toxic mussels now, then we will be infected. Also, if something has eaten those toxic mussels, so say fish eat those mussels and then, well, we eat them, okay? So that's how that is toxic. And then also dinoflagellates or plankton also produce neurotoxin, and we see this in the red tide. So this is going to happen if there's a lot of favorable environment. So it's going to cause rapid reproduction of these dinoflagellates, and they produce these neurotoxins. And that's going to kill all kinds of things. So it's going to kill fish, marine mammals, and humans if we go in the water during a red tide. So those are some of the toxins produced by algae. Moving on to protozoa. So protozoa are unicellular, eukaryotic chemoheterotrophs. They are a very large and diverse group. So different protozoa are going to have very different characteristics. There's only a few that are pathogenic. Rather, there's actually a lot that are quite helpful to humans. We have a lot of protozoa that are part of our normal microbiota also. Protozoa also have very complex life cycles that may involve multiple hosts. Protozoa can be found in the soil and in water, as well as in the normal microbiota of animals. So that could serve as a reservoir as well. So there's lots of different forms. So the first form we're going to talk about is the Trophozoite. This is the vegetative form. So this is the healthy, normal form of the cell, and that's called the Trophozoite. Some protozoa can produce a cyst. So a cyst is kind of like the spore we were talking about. And it's going to provide protection for the protozoa in adverse environmental conditions. Not all of them can produce cysts, but quite a lot of them can, especially ones that are ingested. So the stomach is an example of an adverse environmental condition. It's very acidic. It's hard for the protozoa to survive in the stomach. But some of them can produce cysts. So they're ingested, they produce cysts, so they survive in the stomach, allows them to reach the intestine, where then they will turn back into trophozoites and then be able to reproduce. They can reproduce through asexual reproduction. So they can do fission or budding or schizogony. So schizogony is a specialized form of reproduction that the protozoa undergo. And what happens in schizogony, here's a picture down here. Normally when something reproduces, we have one cell with one nucleus, and that produces 2 cells each with one new nucleus, okay? But in schizogony, instead, the nuclei will divide first, and it's going to divide multiple times before the cytoplasm and the rest of the organelles are going to divide. So here we have one cell, one nucleus, and we see that nucleus has divided again, divided again. We end up having one cell with lots of nuclei in it, and then the cytoplasm will divide so that we eventually have these merozoites. Protozoa can also reproduce sexually through conjugation. Now, protozoa are pretty neat because they are able to move food towards one part of the cell, which is kind of like the mouth, the cytosome. It's going to use cilia to move that food towards the cytosome. And then the amoeba is going to phagocytose that food. The food is digested in the vacuoles, and then the waste will be eliminated through the anal pore. This is all happening in these protozoa. Some examples of medically important protozoa. So Trichomonas is an example of an STD. There is no cyst stage. This is because there really is no need for this species to be able to survive in an adverse condition. Being an STD, it's transmitted directly from a favorable environment to another favorable environment. Symptoms of this disease include itching, burning during urination, and discharge with a foul odor. Giardia is another example. This one causes severe explosive diarrhea. This one does have cysts. Giardia is usually found in contaminated water sources. And so if that's ingested, and then that's going to survive in the stomach through the cysts and cause disease. We also have entomoeba that causes dysentery, also digests or ingests the red blood cells. Acanthamoeba causes eye infection and blindness. Plasmodium causes malaria. We're going to talk more about that one in a minute. Babesia causes hemolytic anemia. Toxoplasma causes a foodborne illness. But where it's really significant is that it causes stillbirth. So pregnant women have to be very careful to not expose themselves to toxoplasma, which can be carried by cats. So that's why they usually tell pregnant women not to clean out the litter box. We also have Cryptosporidium, causes diarrhea, and Trypanosoma can cause coma and death. All right, so Plasmodium vivax. Remember, this is what causes malaria. So we have a vector is going to spread this, usually mosquitoes, and it's going to reproduce sexually in the mosquito. We're going to talk about it more on the next slide, but we are going to have what we call definitive and intermediate hosts. A definitive host is the host in which that species undergoes sexual reproduction. So in this case, the Plasmodium vivax is sexually reproducing inside of the mosquito. So it's the mosquito that's going to serve as the definitive host. Then the mosquito will bite a human and transfer the Plasmodium vivax into the human, where it's going to continue to reproduce, but it's going to reproduce asexually in the human. So the human will serve as the intermediate host. In the human, so we have sporozoites. Sorry, let's back up a step. We have the sporozoites inside of the mosquito here because it's reproducing sexually. Remember, we talked about that on the previous slide. Sexual reproduction is going to produce sporozoites in the mosquito. Then it's going to transfer those sporozoites to the human when it bites them. Then those sporozoites are going to migrate into the liver. And in the liver, it's going to undergo schizogany and produce merozoites. Those merozoites will then move into the red blood cells. And it's going to produce this ring stage. So you can actually see like a ring inside of the red blood cell. It's very definitive. So if you look at red blood cells and you see this ring inside of it, then that indicates malaria in that patient. This ring stage is where the merozoites are continuing to divide inside the red blood cell. Eventually that will cause the red blood cell to burst, spreading those merozoites even more. If a mosquito then bites that person who has malaria, then that mosquito will pick up those merozoites, which will then undergo sexual reproduction, and the cycle will continue. All right, so that's protozoa. Now we're going to move on to helminths. So helminths are parasitic worms. There's two main types. We have the platyhelminths, which are flatworms, and the nematoda, which are roundworms. So platyhelminths, flatworms, nematoda, roundworms. There's two different types of the platyhelminths that we're going to go over. Trematodes are commonly called flukes, and cestodes, which are commonly called tapeworms. Now, all helminths are very specialized to live inside the host, so they cannot survive outside of a host. So we're going to see some of these unique physical characteristics of the helminths that make them very good at being parasites. All helminths are multicellular. Usually they're actually quite large. And they have this specific life cycle. So they're an egg, which hatch into a larvae, and those develop into an adult. So we're going to keep seeing that cycle. We will quite often see definitive and intermediate hosts also with the helminths. Definitive, again, remember, is where a sexual reproduction occurs. And with the helminths, we're going to see that it's the adult stage that carries out sexual reproduction. That makes sense, right? And so this is going to be carried out in the definitive host. The larval stage, we may have some, well, the larval stage is where we're going to see inside of the intermediate host. All right, so platyhelminths. Remember, these are what they call flatworms. And they're called flatworms because they look flat. So they're dorsoventrally flattened. They have actually where their mouth would be is going to be an oral sucker. So it's going to attach to the tissue, the intestinal tissue. Okay, and then they're going to absorb food. They're not going to ingest food through their mouth. Rather, they're going to absorb it through the cuticle covering. So they're just going to absorb it through their surface. And this is the oral sucker here. It's going to latch onto the intestinal wall of the host to prevent that platyhelminth from just being excreted with the feces. Okay, so it wants to stay in the gut. So it's going to do that by attaching to the intestinal wall. So again, I said we were going to talk about trematodes and cestodes. So trematodes are typically called flukes. Cestodes are called tapeworms, and they're called tapeworms because they look like this. They have all these different segments in them. The segments are called proglottids. And it's in the proglottid where we have the like the eggs, I can't think of the word, like the reproduction's occurring, I guess. Okay. All right, here's an example. So this is tapeworms. Taenia soleum and Taenia saginata. So this can occur in pigs and cows. So what we see is we have an infection in a human. We have the tapeworm is in the intestinal gut. It's going to produce eggs. The proglottids are producing eggs. Those eggs are excreted in the feces. where they're then ingested by either a pig or a cow. Then that egg is going to hatch and turn into a larva. The larva is going to bore into the intestinal wall and move into the muscle tissue. Then, once in the muscle tissue, it will form a cyst. Then that cyst in the muscle tissue is then ingested by the human. So when we eat pork or beef that's undercooked, we're going to be eating those cysts that haven't been killed. The cysts survive the stomach, and then they reach the intestine of the human, where then they turn into the adult stage. Okay. So let me just note here that the human is the definitive host because we saw it was the adult stage that was in the human. And it's the adult stage that's producing the eggs, right? And then here, the pigs and the cows are going to serve as the intermediate host. Nematodes, on the other hand, are roundworms. So roundworms are a little bit different. They look round, of course. They also have a complete digestive system and a complete body cavity. The eggs are the infective stage for humans. So the humans are going to be ingesting the eggs. So here is an example of Ascaris. So Ascaris is the second most common worm infection in the United States. And they have a little bit of a different life cycle because we don't have a different species serving as a definitive host. So what happens with the Ascaris infection in humans? So the humans are going to ingest the eggs. They're going to hatch into larvae. Those larvae are then going to migrate into the lungs. So instead of moving into a different species, they're instead going to just move to a different area of the human. So the larvae migrates to the lungs, then they're coughed up and then re-ingested. They cough up and then they swallow it again. And so now the larvae moves down and where it turns into the adult form in the gut, produces eggs, and those eggs are released in the feces where they're ingested probably by a different host. These are pinworms. These are the most common worm infection in the United States. So with the pinworm, this is also another nematode. The female pinworm is going to just a little bit come out of the anus at night while the host is sleeping, and it's going to release the eggs actually on to the surface of the anus. And then those eggs will be removed from the anus with the feces, where it can then be spread and ingested by someone else. So that's pinworms. Last example is the heartworms. So heartworms can be in humans, but they're mostly found in dogs and cats. It's a little bit different here because it's actually spread through mosquitoes. We do see that the heartworms can also cause infection in the human lungs. This is a heart that's full of heartworms. What a disaster, right? It can be very detrimental. And in this case, it's actually the larvae that are the infective stage. All right, so that's helminths. Now we're going to talk about arthropods here at the very end. So we've already talked about arthropods being vectors. Vectors being that it's an insect or an arachnid that's carrying the pathogenic species either inside it or on it. We're going to talk about two different types of arthropods. Include insects. Insects have 6 legs, include lice, fleas, and mosquitoes. And the other type is arachnida, which has 8 legs and includes mites and ticks. Again, they're vectors. If the pathogenic species replicates inside of the vector and it's undergoing sexual reproduction inside the vector, then the vector will serve as a definitive host. What we see is quite often one of the easiest ways to control a vector-borne disease is to control the vector itself. So for example, diseases that are spread by mosquitoes, the best way to control that disease is by eliminating mosquitoes or by reducing the risk of being bitten by a mosquito. So using bug spray, covering up with clothes, having a sleeping net, etc. We've already talked about the different types of vector transmission. So again, remember, mechanical transmission is where it's going to carry the pathogen on the surface of the vector, but it's not actually going to have a part of the life cycle inside of the vector. So that pathogen is not going to replicate inside the vector. It's just being carried on the surface of the vector. Whereas biological transmission is when the pathogenic species is replicating and it does have a part of its life cycle stage inside the vector. Lastly, we're going to talk about scabies. So scabies is a little bit different. So scabies is not carrying a pathogen. Scabies is pathogenic in itself. So in this case, the arthropod is causing disease directly. It's causing disease because it's burrowing in the skin and laying its eggs in the skin directly. So scabies is a mite, and mites burrow in the skin and lay their eggs. This is going to lead to intense itching and this rash that we see here. It's spread by direct skin-to-skin contact. We see this being an issue in places where people are in close contact. So nursing homes, extended care facilities, prisons, where we see a lot of scabies outbreaks, in the hospital too sometimes. They can be diagnosed. Sometimes you can see the mites directly, you can recognize the rash, or you can do a skin scraping to look at them. All right, so that's everything for the eukaryotes. And we're going to talk about viruses next. So I'll see you soon.